Myself, Arsalanur Rahman. I have been teaching biology from past 10 years, almost 10 years, you can say. And I have been teaching biology for class 11th, 12th, and NEET mainly. Okay. Uh, for the demo, I have chosen a very interesting topic or a chapter, that is molecular basis of inheritance. So let me write the name of the chapter, molecular basis of inheritance. First, let me take a shape. Okay. Now let me take a marker, then I'll start writing. Okay, the name of the chapter is molecular basis. Molecular. Sorry for the bad writing, but I'm new to this writing tab. I used to have a different writing pad. Okay, the chapter name is molecular basis of inheritance. Of inheritance. Okay, first we will try to understand. Okay, let me change the marker. First, we will try to understand the meaning of inheritance, and then we will try to understand the whole meaning of molecular basis of inheritance. What is inheritance? You must have seen kid, baby born in a hospital, or you must have seen. If you don't, you must have seen someone said that you look like your mother or you look like your father. That means the characters are transmitted from parents to offspring. That means the character are transmitted from parents to offspring. The process by which character are transmitted from parents to offspring is known as inheritance. You must have studied this in previous chapter. Okay, let me write down for you the process by which characters are characters are transmitted from from parents to offspring. From parents to offspring, it's known as parents to offspring. Offspring is called inheritance. Oh, so you now know the meaning of inheritance. It is a process by which characters are transmitted from parents to offspring. Okay. Now here the chapter is name of the chapter is molecular basis of inheritance. There must be some kind of molecule which is responsible for what is there is some kind of molecule which is responsible for inheritance. So in this chapter we have to study about this molecule only. Okay, in this chapter we have to study about this molecule only. Okay, so what we have to study, molecule which is responsible for inheritance. So what's the name of this molecule? Have you heard about the term nucleic acid? So this I'll let you know, this molecule is known as nucleic acid. So in this chapter we have to study about nucleic acid. Okay, got the point? Okay, have you heard about nucleic acid? In class 11th in biomolecule you must have studied about the nucleic acid, or if not, let me ask you, what is the full form of DNA and what is the full form of RNA? You will say deoxyribonucleic acid. DNA contain nucleic acid, RNA ribonucleic acid. So in this chapter we have to focus on DNA and RNA only. Okay, you already know the full form, so I'm not writing the full form here. Okay, okay, let me explain further. The first topic is history. The first heading you have to write is history. Okay, let me take a shape. Okay, first we have to, in NCRT there is some history given about nucleic acid. So first we have to study about this history. Okay, so first one, the first scientist we have to study, let me change the color. The first scientist is Friedrich Mischer, F. Mischer. The first scientist you have to study is Friedrich Mischer, yeah, F. Mischer. Okay, what you have to know about Friedrich Mischer? Okay, what you have to know? You have to know that Friedrich Mischer was studying about pus cell. Okay, let me change the color. Friedrich Mischer was studying pus cell, or in some book it is written WBC. So let me make a cell for you. And inside nucleus he found dense structure. Inside nucleus, what did he found? He found dense structure, and he called them, what did he call them, this dense structure? Nuclein. What did he call? Nuclein. Okay, okay, I guess I have chosen the wrong pen. Sorry for the inconvenience. Okay, I have chosen the wrong pen. Okay, I'll write down for you one more. Friedrich Mischer was studying pus cell, and he found out inside nucleus there is a dense structure. He found out there is a dense structure inside nucleus. Okay, this dense structure is known as, he called them as nuclein. Called them as nuclein. I don't know why this flickering is happening. Okay, okay, there is another scientist known. Have you heard about Ant-Man? There is another scientist in your syllabus, not Ant-Man, it's Altman. So the second scientist you have to study is Altman. What did Altman do? Altman tried to study the nature of nuclein. Altman studied the nature of nuclein. Nature of nuclein. And what did he found? And he found out, okay, let me change the marker. And he found out the nature of nuclein is acidic. He found out nature of nuclein is acidic in nature. What did he found? He found out the nuclein is acidic in nature. Okay, what can you conclude from this? What can you conclude from this? Okay, what does acidic nature means? What does acidic nature means? Okay, acidic nature means, it means the charge, everything which carry acidic, which have acidic nature, it carry negative charge. So on the the charge on the nuclein is negative. Okay, he found out negative. So the two, you have to remember the two scientists. First scientist is Friedrich Mischer. What does Friedrich Mischer found? He found in pus cell that a dense structure is he's found. And this, he called this dense structure nuclein. And another scientist named Altman tried to study the nature of this nuclein, and he found out the nature of this nuclein is acidic, and this nuclein is found inside nucleus. So the so from here the new name originate, that is nucleic acid, which is found inside nucleus. Okay, so that's the origin of nucleic acid. Okay, now we will talk about now we will talk about now we'll talk about what is nucleic acid. Okay, now we will talk about what is nucleic acid. So nucleic acid is nothing but it is a polymer. It is a what? It is a polymer. Polymer of what? The polymer of nucleotide. Nucleotide. I will use NT, capital N, small t, to denote nucleotide. Okay, okay, let us understand the meaning of polymer first. What is polymer? So polymer is nothing but a long chain made up of, comprising of very small units. Okay, suppose this is a long chain of beads. Okay, So this from there to there, from here to here, this whole structure will be called as polymer. This whole structure will be called as polymer. When we take very small units and when we combine them to make a longer unit, that longer unit will be called as polymer, and the small unit is known as monomer. So this is polymer and this single unit is called monomer. So what is nucleic acid? Nothing. It is a polymer of what? Nucleotide. So you have to add many nucleotide together. You have to add what, Bicha? You have to add many nucleotide together to form a long chain. To form a long chain. Thousands of nucleotide, crores of nucleotides. Okay? In reality, billions of nucleotide to form a long chain, and that long chain will be called as nucleic acid. Either it can be DNA, deoxyribonucleic acid, or it can be RNA ribonucleic acid. Okay, so this polymer is called nucleic acid. This polymer is called nucleic acid. Okay, now let us understand what is nucleotide. Now we have to understand the meaning of nucleotide. Okay, so now we will try to understand what is nucleotide. It is a large molecule made up of three different molecules. Okay, you have three different molecules, A, B, C, and you have to combine all the three and it will form one single nucleotide. And this nucleotide, you have to take thousands, millions of nucleotide and add them together to form nucleic acid, either DNA or RNA. First, you have to make nucleotide, and for making nucleotide you need three different molecules, one, two, and three different molecules. I'll explain you. To make nucleotide, you need three different molecules. First molecule is pentose sugar. First molecule is pentose sugar. The second molecule is nitrogenous base. Nitrogenous base. And the third molecule is, it is an acid known as phosphoric acid, a phosphate group. Phosphate, I'll write phosphate group here. Okay, so start from I'll start from pentose sugar. I'll start from pentose sugar, then we'll move towards nitrogenous base, then we will move towards phosphate group, and together we will try to combine all of this together to form DNA. Okay, let's move forward. So I'll start with pentose sugar. I'll start with pentose sugar. It is of two type. First, deoxyribose. First, deoxyribose. Okay, so I'll show how to make the pentose sugar. Pent means 5 carbon, Ose means sugar. Pent means 5 carbon and Ose means sugar. Okay, so I'll try to make pentose. First, draw a smiley. It represents oxygen. Then make two hands for this smiley. Two hands. Then again make two legs for this smiley, and again make base for the smiley. Okay, every dot represent carbon. 1, 2, 3, 4. These all are carbons. Okay, now what will happen, carbon need 4 bonds. Okay, so here you can see this carbon have 1 and 2 bonds. So make 2 bond here, 1, 2, make another 2 bond here, make another 2 bond here, another make 2 bond here. How many carbon can you see? This is 1, carbon number 2, carbon number 3, carbon number 4. You can see 4 carbon. So I have already told you we are making 5 carbon sugar. Okay, so you just make 1 carbon CH2OH here. Okay, let me erase. Okay, this is the 5th carbon. Okay, this carbon required 3 more bond, so two bond is CH2OH. This will be CH2OH. This one will be H. What will do? Just add HOH simultaneously. HOH, HOH, HOH. I'm not taking organic class, so you can make HOH anywhere. And here H. How many carbon can you see here? You can count 1, 2, 3, 4, 5. 5 carbon. You can count 10 hydrogen, and you can count 5 oxygen. So this is the formula of what? Deoxyribose sugar. So this is deoxyribose sugar. This is deoxyribose sugar. Okay, now we will make another sugar just like this, but what is the we will try to make another sugar name ribose. Okay, sorry, my bad. We have make ribose sugar here. We have make ribose sugar. Okay, this C5H10O5 is ribose sugar. My mistake. That was my mistake. Okay, now we will try to make deoxyribose sugar. What? Deoxyribose sugar. Okay. So how to make deoxyribose sugar? Here you can say D. D means removal. D means removal, or oxy means oxygen. So removal of oxygen. You have to oxygen remove oxygen from where? At carbon 2, carbon number 2. So again we will make same smiley, 2 hands, 2 legs, and a base. Make 2 bonds everywhere. Make carbon number 5 CH2OH, add hydrogen, and now add HOH. But at carbon number 2, here you can see it OH is present, extra oxygen is present. Now you have to remove oxygen from here, so just add hydrogen. Bond will be same. Okay, उसके बाद you have to add again HOH. So this is what is the formula of this sugar? C5H10, and here you just remove one oxygen only, so you just have to write O4. This is ribose sugar. This is ribose sugar. Now tell me, out of these two sugar, which sugar is more reactive, or which sugar is more stable? Okay, so C C5H10O5. This is ribose sugar. It will be present in RNA, and this is C5H10O4. This is deoxyribose sugar. This will be present in DNA. Here, the number of carbon and the hydrogen is same. The number of carbon and the hydrogen is same. So the electron contributing by the carbon and hydrogen will be same. Here there is one extra oxygen means 2 extra lone pair, that is 4 extra electron, and you know the reactivity depends on number of electrons. So this will be more reactive, this will be more reactive, or this will be less reactive. Now you also know, more reactive the substance is, yeah, compound is, or a molecule is, the less what stable it will be. Okay, so this is less reactive, so it will be more stable. Here you can see the DNA sugar is more stable. That's why DNA is overall more stable than RNA. Now here this will be less stable. That's all for the demo lecture. Thank you.